trick. So you are welcome and please continue. Over to you.
Okay, thank you very much for giving me an opportunity to deliver a lecture on recent advances in plant virus disease diagnostics, where we can have a case study on apple viruses, how we are diagnosing these apple viruses. So first of all, might be knowing that what is actually a disease. When we say what is disease, this is a harmful deviation from any normal physiological process. When we have any deviation, when we feel sometimes that something is happening to our body or plant, sometimes any kind of symptom is there, any kind of expression is there, we may call it as a diseased plant or diseased human being or diseased animal. So which disrupts the plant's normal structure, growth, function, and other activities. For example, you might have seen in your fields that how plants are behaving when there is a disease. Their growth gets retarded, their photosynthesis gets reduced, the quality of the fruit, the quality of the produce is not up to the mark. So when such things are happening in any plant or in any human being or animal, we call it as a disease. Dis and ease, it is a French word actually, which means that away from the ease. When somebody feels away from the ease, then we call it as a disease. So similarly, there are number of pathogens which are causing these diseases. Pathogen is an entity which causes the disease, which is responsible for the cause of the disease. And among the pathogens, we are having the fungus, we are having the bacteria, we are having the viruses, we are having the phytoplasma, protozoans, algae. Number of pathogens are there which are causing the disease in plant, animal, and human. So among them, very important entity that is the virus. Uh, Everybody is knowing that how this virus has created a havoc in 2020, that COVID-19, the very important and famous disease which we encountered in last 2-3 years. So virus is a very important pathogen, and it is actually only the nucleic acid and having some code protein, having a protein shell around it. Just it is a game of nucleic acid only, and it is encased in the code protein. But if we see other pathogens like bacteria, fungus, they are also submicroscopic particles like bacteria. But virus is very tiny particle, very small genome, very small size as compared to bacteria and fungus. It's a very small entity which causes the diseases, whether in human, whether in animals or in plants. So we are dealing here with the plants. We'll see how it's causing the disease in the plants. Here you can see one of the images there. Here the nucleic acid can be either DNA or RNA. Just like in humans, we are having both RNA as well as DNA. But in viruses, it is only either it can be a DNA virus or it can be a RNA virus. For example, what the COVID virus was, it was a RNA virus. We are having the only RNA. If, for example, another virus is that in humans it is causing the AIDS, that is a DNA virus. So it can be either RNA, it can be either DNA. Similarly, in the plants also, it can be either DNA or RNA virus, which is causing the disease. And one more photograph is there. It is a tulip breaking. Actually, this tulip breaking disease has given the birth to the virology in plants, that the viruses are also causing the disease in plants as well. So this is one tulip flower which was actually infected with the virus, tulip mosaic virus. And but that time they were seeing this tulip as a very beautiful flower with this tulip mosaic virus. But later it was confirmed that it's the virus which is causing this type of the symptom. So these are the different types of symptoms which are caused, these viruses are causing in the plants. You can see different types of symptoms like the first it is a mosaic type of symptom, then it is a curling symptom, then it is a pitting symptom. We have another crinkling symptom, we have anations, we have little leaf, we have curling. So n number of symptoms are caused by these viruses. And one more thing in these viruses is that sometimes it is a latent infection. Latent means when it is not expressing any kind of symptom. I will give you example when sometimes we went for the COVID testing. We were not having any kind of the symptom, but we were found positive for the virus. So similarly, sometimes these viruses only cause, they are not causing any kind of the symptom, but they will remain in the plant or they will remain in the human, not causing any symptom. Then we call this infection as a latent infection. So in plants also, and in plants, especially in the perennial crops, these latent infections are more as compared to the infections which are causing the symptoms. So now, when these viruses, because we don't have as such the management, just like we have for the fungus, like fungicides, and for the bacteria, like we have antibiotics. But as far as these viruses, we don't have the viricides as such, which controls these viruses in the plants. So ultimately, what we have to do detect these viruses in the earlier stages. And when we detect these viruses in the earlier stages, then we can have the plants which are virus. So we can, if for example, we have a plant which is not which is virus infected, then we have protocols by which we can make that plant virus free. Like we can use the tissue culture technique, where we can use the cryotherapy, thermotherapy, or some other therapies by which we can the virus, we can remove the virus from those plants, and those plants will be cleaned and multiplied and supplied to the farmers. So we should have a very robust diagnostics. We should have very strong diagnostics to detect these viruses, especially in our perennial crops. So for the detection methods, when we have to detect any the virus or any pathogen, some important prerequisites are there for detecting any kind of a pathogen. So first and foremost is the specificity. That whatever virus we are target, whatever pathogen we are target, we should able to detect that. The specificity means the ability to distinguish the target pathogen from all the organisms. For example, in case of these COVID viruses, there are n number of COVID viruses. Sometimes we might get the COVID infection, but it can be some other type of coronavirus, which is not actually the coronavirus which is causing the COVID disease in humans. So there should be specificity. That is why you might, I will tell you about the different diagnostic techniques. Why in COVID testing also that the rapid antigen testing was not as perfect. What was perfect was more the real-time PCR based or RT-PCR based. So there are different methods that those methods should have the specificity to target that pathogen which we want to target. Then another is the sensitivity. Sensitivity means up to which mark we can detect that virus. The ability to detect target organisms which are potentially significant in field crops or even perennial crops, even at low incidence. For example, if we see the viruses in our perennial crops like temperate fruits like apple, peach, apricot, plum, the virus titer varies with respect to the temperature. So, for example, in spring, if the virus titer means the concentration of the virus, we can simply learn this. If the virus is very high, some methods can detect. So that means, for example, the ELISA will detect. But if we see that virus in summer months, the ELISA cannot detect. Why? Because the titer gets very less. So when titer gets less, the sensitivity of the technique gets correlated like that only. ELISA has a less sensitivity as compared to the PCR. So PCR can be used to detect the virus in, for example, in the summer month. The ELISA, for example, has the sensitivity up to 10 raised power minus 10 particles. But if we see the PCR, it is having the this sensitivity up to 10 raised power minus 17. Minus 17 means very less because it is minus 17, 1 by 10 raised power 17. So the sensitivity is very important because, for example, if the titer is very less, we are going for any kind of detection method, but it will not detect. So we'll say it is a virus free. But actually, the titer is very less. We couldn't detect it with one technique, so that the results will be wrong. So the specificity is very important. Sensitivity is very important. Then the speed. That our technique, it should not take 3 or 4 or 7 or 10 days to detect any kind of pathogen or any kind of the virus. So the speed is very important. Accuracy is very important. Then the simplicity. Simplicity means that we should not have the complex type of detection methods where we have to use very technical manpower, we have to use sophisticated machines, we have to use sophisticated chemicals. So the technique should be simple. The method should be simple where, by which we can detect the virus. Then obviously the cost effectiveness, for example, you are seeing in the COVID times, the kit, that rapid antigen testing kit, which might cost 100 rupees. There are other methods, for example, real time, it will take 500 rupees. So we have to develop such kind of techniques, such kind of methods, which are very easy, which are very sensitive, which are having very this robustness, simple, but at the same time, it should be cost effective. And especially in the plants, for example, we have to test the population. We have to test the thousands of plants. So we should have those testing kits which are cost effective. We should not give more burden to our farmers or any research organization to have such sophisticated techniques. Then another is the reliability. Reliability means it should have sufficiently robust so that the results are repeatable between the labs, between the samples, or between the companies. So this technique, those techniques should be reliable, where we can have the trust that whatever it has detected, it has detected. It is true to the nature. So these are the various characteristics of any detection method. Whether we have ELISA based, we have protein based, we have nucleic acid based, we have physical property based. So these methods should, these properties should be here to any detection method. But we have n number of detection methods, which I will explain in later slides. Maybe some may be having two, some may be having three, some may be having one. But all these methods may not be efficient as far as these properties are concerned. But yes, we should look for those methods which have at least the maximum number of these properties. So these are the various methods based on which we can detect these viruses. One is the biological property based, like we can see the symptom and we'll tell this kind of virus will be infecting this crop. Our transmission test, we have various types of transmissions, like mechanical, like vector based, and we can detect or we can tell that this virus might be this. Then second is physical property based, like we can use the electron microscope to detect these viruses. And we can use some light microscopy in some cases of viruses like potiviruses, where we have inclusion bodies specific to those viruses, and those can be seen through light microscope, but that is not very common. Electron microscope is very important as far as by observing these viruses. But very important and very useful techniques are based on the protein property, and one is based on the nucleic acid property. Because these viruses, if we see their structure, it is composed of protein, that is the code protein around the nucleic acid. So there are two quantities, nucleic acid and the protein. Those can be used for detection of the virus. For example, if we see the protein based, we can see the ELISA, very important enzyme linked immunosorbent assay, where we are using the antibodies directly. We are developing the antibody against any virus, and then we are seeing the color changes when the virus is present. We can see the color change, and we can tell that virus is present or not. But yes, protein based this detection is having because of the less sensitivity of these antibodies, because we are mostly using the polyclonal antibodies for detection. And so this is again having some drawbacks regarding the sensitivity or the specificity of the virus. And very important among them is now the nucleic acid based detection. So we have n number of methods in the nucleic acid based detection. Nucleic acid means the composition, whether RNA or DNA. So anything from the RNA or DNA which we are using for the detection, that we call as the nucleic acid based detection. We have number of methods. You can see here, nucleic acid spot hybridization is there, polymerase chain reaction is there. These are variants of the this polymerase chain reaction, like RT-PCR, then cooperational PCR, multiplex PCR, nested PCR, real-time or quantitative PCR. And one more very important nowadays, which are being used, that is called the high throughput sequencing or next generation sequencing. So first and foremost important, which we are using for characterizing or for detecting the apple viruses, is the PCR, the normal PCR, polymerase chain reaction. So first, if we know that the plants are infected with the RNA virus or DNA virus. So accordingly, we will go for the isolation, either DNA or RNA. And if we will go for the RNA isolation, then we will go for the cDNA synthesis. After cDNA synthesis, we go for the gene-specific amplification or gene-specific primers. So mostly in viruses, there are three important genes. we are called as the open reading frames. One is the code protein gene, another is the movement protein gene, and third one is replicase. So these are the three important genes present in the virus. One is code protein gene, which codes for the code protein. Another is movement protein, which helps this virus to go for the movement in the plant. Then third is the replicase. The replicase is the enzyme which helps in replication of that virus or the RNA or the DNA. So these are the three genes, and these three genes are utilized for developing the or designing the primers. When we design the primer, then we amplify the primer, this virus in PCR. You can see here we have this apple necrotic mosaic virus. We have amplified it as code protein 670 base pair. Then we cloned that in a vector. After cloning, we went for the sequencing of that 70 base pair. After sequencing, we went for the blast analysis. And after blast analysis, you can see what we observed, that is the apple necrotic mosaic virus. So this is a virus which we have identified using the PCR-based technique. And we are using now this PCR-based technique for detecting this apple necrotic mosaic virus or apple mosaic virus or other viruses which are infecting our apple crop. So next method which we developed, for example, these perennial crops are infected with many viruses. We are calling them the storehouses of the viruses, where n number of viruses are present, are infecting these crops. So when, for example, if we have to go for the single PCR and single detection of the virus, and for example, we have 4 or 5 viruses infecting the apple crop, then we have to go for the individual virus amplification, individual PCR we have to go. Then we will detect the 4 viruses in single, not in single, but 4 times we have to put the PCR and we have to detect it. But there is a method we have developed, that multiplex PCR, that in single go, in single amplification, we are detecting all the 4 viruses. For example, you are seeing here, there are 4 viruses: apple necrotic mosaic virus, apple mosaic virus, apple stem beating virus, apple stem growing virus. So here you can see the single, and single. But in one tube, we are detecting, when we are detecting single, in a single tube, all the 4 viruses, we call it as a multiplex PCR. Some, if we will detect 2 viruses at a time, we can go call it a duplex PCR. When we go for the 3 viruses at a time, we can go for call it a triplex PCR, quadruplex PCR, pentaplex PCR. So it depends upon how many viruses or how many pathogens are we detecting simultaneously. So here the advantage is that we are reducing our time component, we are reducing our cost component, we are reducing our this management component. So this multiplex PCR is more efficient than the normal PCR when we are detecting the single virus. So this is the validation that, for example, if we have different varieties, for example, we have 7 varieties here where we have validated this multiplex PCR. Means if we have to detect the 4 viruses, we have to go for 7 into 4, that is 28 PCRs we have to go. But in only 1 or 7 PCRs, you can see that we have detected 3 viruses, we have detected some of the varieties 4 viruses. So this multiplex PCR is having this advantage that it is cost effective, it is time effective, and we can manage the things and we can reduce the time of the for the detection of the virus. Otherwise, for example, for the normal PCR, what we have to do, first day we have to go for the RNA isolation, then we have to go for the cDNA synthesis, then we have to go for the PCR amplification, then we have to go for the gel electrophoresis. So that much activity we need for the single virus. But if that thing is taken for the 4 viruses, obviously we have reduced the time from 4 to 1. Similarly, for the real-time PCR, and real-time PCR is a very important, I can say, the extension of the normal PCR, where we can detect the virus in the real time. Real time means, for example, we don't need any kind of the gel electrophoresis. Here you can see these curves. When it starts to curve, when it starts to showing the upward curve, we can see that time only that this virus is present. So when it starts early, we can say the titer is high. When it starts late, we can say the titer is very high. We don't need any gel electrophoresis in this. But yes, these PCR methods, very important part in this PCR method. We need a high sophisticated machine. Almost it costs us 3 lakh, 4 lakh, or 5 lakh rupees. Even real-time PCR machine costs us 14, 15 lakh. So we need huge equipment for this. We need costly equipment for this. But yes, sensitivity, specificity is very nice and very high in these methods, but they might not be cost effective. They might not be as this simple as we observe in the these ELISA methods. But yes, as far as the sensitivity is concerned, specificity is concerned, they are very this advanced methods as compared to protein-based methods. So now the another approach has come that, for example, if we have a plant, we don't know what are the viruses or which type of disease it is infected with. So we have now the approach that is called a high throughput sequencing or next generation sequencing. So we don't need any kind of information before that. For PCR or for ELISA, we need information, prior information that which virus might be. So accordingly, we go for the designing of the primers and then amplification. But here we have a very robust approach. We don't need any kind of information earlier. Just we need infected sample, and then we need approach. That which type of approach or which method we need to utilize, or which method which we will go for the sequencing and identification of the viruses. We have approaches like RNA sequencing. RNA sequencing is nothing but we sequence a whole RNA. For example, if we take the Apple example, we give the sample to the company, they will take the RNA and they will sequence whole RNA. And then from the RNA sequence, whatever sequences they have come from that RNA sequencing, the Apple part is removed through the softwares. Apple part is removed. Apple genome sequences are removed. Whatever will be there, then subtracting like that. For example, we have an Apple sample. I will show you here. We were having these Apple samples. One was the asymptomatic, two were the symptomatic. So what we did, we sent these samples for the next generation sequencing. So these are the steps. First, they will go for the RNA isolation, cDNA synthesis, then library preparation. Then they will go for the sequencing, and they will sequence each and every component. Whatever RNA they will get, the plant RNA, the microbiome RNA, whatever microbes will be there, they will sequence each and everything. After that, for example, what they will do, the RNA from the plant part, they will remove. The sequences from the plant part, they will remove. Then what we will be identifying, we have to go for the virus identification. We don't need the fungal part, we don't need the bacterial part, we don't need the microzoa part, we don't need the others. So one by one, we are subtracting everything from that sequence. And lastly, what we are seeing there, when those viruses and virides are there, then we will go for the match with the NCBI BLAST, and we could find, or we could identify, these are the viruses and these are the virides which are present in this sample. So then, for example, we have submitted these samples for the next generation sequencing. You can see the variety asymptomatic, where we could not have any kind of symptom, but we could observe using these next generation sequencing tools. We have observed 3 viruses were present in that asymptomatic plant also. So might be these asymptomatic plants where the viruses are latent, and mostly this apple chlorotic leaf spot virus and apple stem beating virus are latent viruses. They are not expressing any kind of symptom. But this apple green clinical associated virus, which we found first time in our crop, and we were observing the fruit symptoms. I can show you in the first slide. We'll go back. This type of symptom which we are observing in the fruit. This is the symptom. This is the fruit from that plant where we observed this apple green crinkle associated virus. Earlier we were thinking this might be some deficiency, this and that, but when we did this next generation thing, we could observe, no, this is actually this apple green crinkle virus, this crinkling type or deformation of this fruit has happened. So this virus was first time identified using this next generation sequencing. So that is why I was saying that we don't need any kind of pre-information. We just need any kind of infected sample. We'll go for the sequencing, next generation sequencing, we can observe. And for example, you can see the symptomatic part. We observe apple necrotic mosaic virus. We observe apple stem growing virus. We observe apple stem pitting virus. Similarly, at the same time, we observe one viride also, that is apple hammerhead virus. So here we observed another symptomatic. We observed apple necrotic mosaic virus, apple stem growing virus, apple stem pitting virus, and apple hammerhead virus. Otherwise, we could not have the information about this virus if we would not have this next generation sequencing. So this next generation sequencing has revolutionized this plant virology, or I can say this plant pathology, by observing for example, where we are not knowing the etiology of the disease, but with the help of this next generation sequencing, we could delineate, we could detect, or we could identify the causal agents of those diseases. But obviously, after, because this is whole the sequencing, then we also validating these viruses, whether actually these viruses are present in these samples or not. Then similarly, again we go back on the PCR or real-time PCR amplification. But what I was talking about, that we need the huge equipments, we need huge machinery for carrying out these detection methods like PCR, and for this next generation sequencing, we need next gen those machines like big machines we need for this. So another method came, those are called isothermal amplification, where we need a small temperature, single temperature or some time where we can detect these viruses. But every method has its own advantages, every method has its own disadvantages. These isothermal amplifications are now being frequently used. These are called point of care detection. So, for example, we have to test the nursery plants. How we should take these PCR machines and other things there. That is not possible. So these POCD or point of care diagnostic methods, we develop the lateral flow assays, just like we have the kit for this coronavirus. Similarly, we are developing the kits, small kits, these lateral flow assays for these viruses, for the detection of these viruses, and we can take that kit anywhere. We can put the sample. If it gives the color, we can tell, yes, this plant is infected with this virus. If it is not giving color, we can say this virus is not infected, this plant is not infected with this virus. So one of the very important method in this isothermal amplification is LAMP method. That is a loop mediated isothermal amplification. Isothermal means the one temperature. At one temperature, it can detect, like in PCR, we have to keep 4 temperatures or 5 temperatures there. And we don't need any kind of big equipment. Similar small kind of water bath will help us there. And one more this benefit in this method is that it is a very sensitive and specific method, because we need only some the primer, small type of kit, chemical type of kit, and we can detect like that. For example, here you can see we have isolated the RNA. Then we used this lamp mixture, RP lamp mixture, and put our primers. We designed the primers, and all we in this primer, 3 sets of primers are being used, and one polymerase is being used. That is called the strands, this binding strand stabilizing polymerase. All those things are put in one tube, and our template is there. We have to put. Then we can you can see here. The virus, if our sample is not infected, it will not give any color. But if it is infected, it will give color. But here you can see here we have to go for the RNA isolation. But now we are standardizing it that we should take the SAP only, not the RNA. When we are taking the SAP, putting in this tube with the help of this RT lamp mixture, it will give us the color. So that process is being standardized, and that will be taken into the lateral flow assay, where it can give the color or not color. If it will give color, similarly the principle is same. It will give color, it is a positive. If it will not give color, it is a negative. And we have to incubate it here for the 60 degrees Celsius for 35 minutes. But when we go for the lateral flow assay for those strips, it will take just 10 minutes, and in 10 minutes it will give us the color. Similarly, there is one more method which we have developed in our lab also, that is recombinase polymerase amplification. First, we have to standardize it from the nucleic acid. We will isolate the nucleic acid. Then from that nucleic acid, we will put that nucleic acid in small type of a kit. Here you can see the 42 degrees Celsius, but you can detect it from 37 to 42 degrees Celsius. How it differs from the lab? First, that we need 65 temperature there, but here we need 37 degrees. So 38, 32, 39, and that is normal. 37 is normal body temperature. If we have a tube with the SAP and with this mixture of this RPA, we can put in our body for 10 minutes, and it will show the color. We don't need any kind of water bath. We don't need any kind of isothermal device, but simple that kit will be having with us. We'll put those chemicals, we'll put the sap, and putting in the our body for 10 minutes, for 5 to 10 minutes. If it changes its color or if it gives the turbidity, we can see, we can tell that this plant is infected with virus or not. So this method we are developing in our lab, RPA based recombinase polymerase amplification. And one more benefit of this, we can, for example, if we are going for other basic studies, we can clone the gene here. But as far as the lab is concerned, we can't clone. Why? Because in LAMP, we are getting the ladder type of amplification. But in LAMP, this RPA, we are getting the single band amplification. We can take this band, we can clone this band, and we can use this for the further research and other things. So these are the methods. Similarly, this RPA can be taken into the lateral flow assay, whatever scripts we are using for the COVID detection and other things. And those things are in the process, and we are also developing those scripts where we can detect this apple necrotic mosaic virus in the field as well with the help of those scripts. So this is all that how up to which it can detect the virus. For example, you can see here up to 10 raised power -6, it can give our amplification. Even up to 10 raised power -7, or then it is a validation in different varieties, how we can validate this. It has been validated also. So these were the various methods which we have developed for detection of the apple viruses. We have developed also the protein-based methods also, but I have discussed only the recent ones like this isothermal amplification, like LAMP, RPA, and then obviously the high throughput sequencing, because high throughput sequencing is a very important tool where we can have a complete virome of the plant, that how many viruses, how many viruses are actually infecting this crop, and both which are economically important or which are latent in the plant, which are otherwise very difficult to detect through other approaches. So thank you very much for your attention, and any question from yourself, I'll be happy to answer those questions. So if no question is, then
If participants, any questions, please ask, sir. I think, sir, there are no questions.
okay, thank you organizers for giving me opportunity to deliver the talk on apple viruses. Thank you very much, ma'am.
Sajad sir, I am having one question, please.
Say.
Sir, you told about isothermal detection, na? What it is, what you do in isothermal, just I wanted to know.